Thank you very much, Doron, for agreeing to give a Build-a-Cell seminar, and you can get started.
Should I answer?
Yes, it's all you now.
Thank you for inviting me. Oh, so I'm starting.
Yes, we're
This
rolling.
meeting is being recorded. Okay, good. Okay, so I have to start here. My scientific life along ages 11 to 77. Next month. School, a chemistry laboratory at home. BSc, Hebrew University of Jerusalem, major chemistry. Added biology, physics, geology, mathematics, and computing. PhD at Weizmann Institute, chemical immunology. That's an interesting mix. Postdocs at Harvard, immune proteins and Yale olfaction. Early professor at Weizmann, olfactory genetics. Later professor, head of genome and bioinformatics center. And later 25 years at Weizmann, investigating origin of life. The sum of all the above enables the lecture today. Okay, this is the title, and you see that I felt a bit uneasy, but I could have corrected it a little bit. Instead of build a protocell, watch a protocell, because I don't do any experiments, so I can only think about and watch and think and do together all sorts of things. Although I'm an experimentalist originally for many other things. Okay, so I understand what experimentalization is. Okay, so, well, we are talking here about, why don't I go here? Because it's the same thing. So as we all know, carbonaceous meteorites come into our Earth for many, many years, 4.5 billion years actually, since it got here. I say Terra got here. And when you do very clear analysis, very, very accurate analysis, as Philip Schmidt-Coplin is doing, he found 15,000 atom compositions, which means something like C5H7O5. But each of them can be many, many structures. So there may be up to 1 million molecules in the soup. I don't know why for dozens of years people tried to make all of these, like I make alanine by Miller-Urey, I make RNA on clay, I make this and that with HCN to adenine. That's not necessary. We have all the molecules we need to start life. Now, I put this here. Immediately, Rohit asked me whether I stole this from you, but I said, no, I found it on the internet. Is it right? This is yours, Kate.
Yes, I think that's
it's something like talk at the night or something like that.
Yes, I remember that slide. You're right. Huh.
So I said I have to show what you are doing so that I compare it to what I'm doing. So the idea is that, yes, you use all of that, constructing synthetic lipid vesicles as compartment, selecting diverse present life components, and artificially loading them into the vesicle in a bottom up approach, and expecting revolution in biotechnology and attaining better understanding of biology. But I know that all what you're doing, Kate, is actually containing also the interest of understanding origin of life better. So that's really nice. I mean, for both of us. Okay. For some reason, it doesn't go down. Oh, okay. So what we are going to do today is to look at this. Lipids, we do. Small molecules, we do. But we don't touch nucleic acids, translation systems, which is even worse. I mean, you can do RNA, very mixed, of course, with a lot of sequences on a mineral. But to make translation system, that's a completely different story. And proteins, I say this is for later time, maybe 300 million years later than when I am in this lecture. Okay, so what we say here, lipids, other small organic compounds spontaneously form organic protocells. This is easy, we'll see it immediately. And then the organic repertoire involves may well be much larger than the life repertoire. It's funny that most of the papers on how we get a particular molecule it brings you to only look at what is in life today. But there may have been a million molecules, and we don't have so many. We have many fewer. So particularly for bacteria. So this is just a comment. Could protocells progress in bottom-top manner from simple to complex? Okay, comments. My cells play compositional information. It has nothing else. It doesn't have a sequence. So it's a different world of information, and but it has straightforward copying, as you will see. Not very straightforward, but much more than if you tell me I take RNA in buffer and I add all sorts of things, not, of course, enzymes, and you see what happens. Do you get it reproduced? It will not. Okay, how about composition? Most of what we do in biology is composition. Transcriptome is composition. Proteome is composition. Metabolome is composition. How much of each metabolite, and so on. And when cell today has to make another copy, they are very careful about the composition. Of course, they also care about the DNA, which is then transcribed to RNA and translated to protein and so on. But basically, it's very important. And a lot of that is actually epigenetics. Self-reproduction of vesicles with only one type of lipid. As you see below the comment, most of the people do experiments with only one type. Not only that, only fatty acids. Why? Because that's what Miller-Urey type experiments can make in their experiments that try to look like what happened on Earth very early time. So that's wrong. It's wrong to use only one. It's also wrong to do only fatty acids, which is so. Okay. Maybe there is a good chance there were no phospholipids, despite all of these million compounds, but who knows? Okay, let's continue. So production becomes relevant when the vesicle membrane is variegated. That is, beforehand, it's not interesting. Just all yellow to all yellow, you don't have to do anything about that. But if you have to do all of these many colors in different proportions, you have to be very careful how to do that. That can be done. Okay, and all that I will tell you by the end of the day is, or by the night, is for the you is day, for me is night. The idea is that I'm not going to I'm not going to try to do anything that doesn't make sense, but I will not be able to show you. I have quite a few experiments, but not mine. Okay, homeostatic growth is preserving composition upon swelling, responsible for self-reproduction. This actually, since you have to grow, it's homeostatic growth. Okay, so homeostatic means that the composition is the same. It's not the same concentrations, but it is the same ratios. And what is the black is not very important. Most people think that what happens in split is what important in making new babies, but that's wrong. It's the growth that does. Okay, these are just examples, little examples of what happens. You have all sorts of colors in I'm looking at membranes alone. There is not, let's assume for the moment that there is only water inside and nothing else. Okay, but there are many different compounds and organocatalysts, things that not too many people in Origin of Life look at because they always think about enzymes, despite the fact that there were no enzymes to begin with. But that's fun. So there is a blue organocatalyst and there is a red that needs to go in. And this is something that can also be catalyzed, not only covalent, making new bonds, but also non-covalent, which is stick this molecule into the bilayer. But it can be catalyzed. Okay? And the same is true for synthesis. So there is a head, red, and there is a tiny thing that you see inside the membrane. That's a tail that always goes there because it's very hydrophobic, and it can then covalently be bound each to the other, getting the result of, instead of one red, you have two reds. Now we are the real story. Self-reproduction of a protocell with heterogeneous membrane along with populated lumen is even more difficult. I will just say something very simple. You need, no matter what, if I go back, this is metabolism. So you need metabolism, but very broad. The only solution is a single metabolic catalytic network that governs compositional homeostatic growth of both phases, the bilayer and the watery lumen. Now NASA, there are so many questions about this, but I believe they are right. Life is a reproducing, they sometimes use another word, but doesn't matter, it's almost the same, chemical system capable of Darwinian evolution. Most people just completely abandon it because you look, 95% of the papers say no Darwinian evolution instead, if there is no RNA and or DNA. That is absolutely wrong. This means that I say, this black in red says, this means that a very simple chemical system, if reproducing, self-reproducing, and capable of Darwinian evolution, that is actually a result of that, it is alive. So people always say, when does life begin? And they never know how to say that. And I say, it's very simple. It reproduces, that's all. That's interesting. Okay, and that's something that I take most of the picture here is from people that I really admire, Cornish Bowden and Cardenias. And this is what I say. Life begins with when we pass what NASA says is the limit. So it must be and can be very simple. And it's also funny because people don't know what is origin of life, and they work about it and call about this. But the first thing that passes NASA is origin of life. That's it. We don't have to worry about that. And I say that on the way to Luca is probably 500 million years, as I said already, and RNA appeared somewhere on the way. By the way, there are not only one Luca, but this is another lecture. There must have been many, many more. But okay, and RNA should be very near to Luca on the track because it's very, very complex. And RNA alone, as said already, is not enough. You need ribosomes, tRNAs, and so on and so forth. So it must be very near to what we call Luca, which is what a bacterium like today looks like. I also want to speak about pillars for one moment. 3 pillars most times, sometimes 4, never mind. I say there are no pillars, there is only one. There is only metabolism. People always say, this guy or lad and whatever thinks that life began with metabolism. And they say, no, 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 it began with lipids. Oh, no, 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 it has to start with RNA, and so on and so forth. I say, no, everything is metabolism. The container is metabolism. I already said that. And DNA and RNA is also, and everything else. So there is only metabolism. But of course, you have to say metabolism can make membranes, and it can make all sorts of other things, and it can end up, if we go back in time, where am I? It maybe 200 million years beforehand, or maybe 150 million before today, it was when RNA arose, and then there was RNA. So I say, don't worry about these words, just metabolism is important and easy to understand. Back to serious difficulties, synchronous with both membrane and lumen. Only few attempts exist for resolving the challenge of synchronization between the two phases. There are quite a few papers, but not too many that show exactly what we can do about this. I don't know for sure, but I can have some ideas. Many of those involve mutual stabilization, such as, for example, RNA, when it touches the lipid of the vesicle, it becomes stabler, or vice versa. This is not really synchronization. It has to be much more complex than that. This is an interesting paper. Is he with you, Mortas, from Italy? I mean, in the, can you speak? Never mind, don't.
Yeah.
Oh. Is he one of the people that listens to me? No,
I don't
he
see him
does
on
something
there.
very simple, very, very similar. Because what you see, he makes here a vesicle with a phospholipid. And then he adds a complex, which is called E. It's a bacterial multi-enzyme complex to make fatty acids. This is really funny, but this is it. So it's funny that you need all of that, but that's because that's evolution. It came out to be that in some animals, you need something very complex to make certain fatty acids. Okay, so this is wrong. And the reaction, he makes a lipid that is different. So the composition of the lipid is wrong. It's not preserved. And the reaction is also very complex. So I don't know how he says this is interesting for origin of life, but it is to some degree. I don't say anything negative. I just want to say something more positive. And this is positive. It so happens that this is really, really nice for me. And it's not just because I want to be nice to you, Adam, Kate. I just think that it is really nice. So we are talking about a case in which you have two fatty acids with some changes, and you have a table with oh my God, peptide, and this dipeptide is actually made with one catalyst. In this case, another two amino acids, serrase peptide. Okay, but once you have the blue, it gets into the membrane, and what does it do? That's really interesting. It helps lipids to come in. So this is actually a catalyst. Most people say this is not catalyst because catalyst always does covalent bonds, but that's wrong. There are many cases like that, even today. So anyway, so that's really, really nice, Kate. To better understand the two-phase reaction, it is worthy to first analyze only one phase. Okay, so the few next slides, nothing in. I did it already before. There is only water, but what do you do? And that's not easy. But it can be done on micelles, which are much smaller, 10,000 times less lipid, only 100 for micelle or something like that, and millions in typical vesicle. Okay, so anyway, what to do? How do you actually make sure that there is homeostatic growth and the composition is preserved. Okay, now we begin to do, I see here we begin the importance of computational chemistry. And I really feel that this is a very important kind of science. So what do? We have these four. Blue catalyzes, I'm saying catalysis is always for getting in. So blue catalyzes red, red, light blue, green is autocatalytic. It's okay sometimes, but it doesn't have to be. Most people think that it should be. Everybody should be an autocatalyst. This is very, very terrible. They can be sometimes, but not always. And then there is a matrix that shows how strong is what is the strength of catalysis. And that's something that we use a lot in our computational chemistry. Okay, so the matrix of mutually catalytic strength and the lipid composition of the vesicle membranes will determine whether the membrane will undergo compositional homeostatic growth. So give me the matrix. I can actually compute out what is the composition. That the composition is something that copies itself in the stories that I'm talking about. So anyway, important to know what is what numbers to put in the micelle. Sorry, in the micelle. Oh my God, I'm very funny. It's a matrix. Okay, so in the matrix, I say the following. I have to think what is the distribution. So the distribution, because of my paper from 1993 about what happens in the immune system, this was what we found out with experiments, not us, others people, that actually it's always log normal for how prevalent is an affinity that is big, medium, or small. You see that it is very many weaks and very few strong. It's the same for catalysis. And I will say it again next few sentences, but if it doesn't be like that, we showed by computing, you don't get reproduction. You don't get it. Reproduction requires this. And the second is what you begin with. And if you begin with, it probably find its way to the reproduction. You'll hear more about it. But you can actually, if you have the numbers of the distribution, if you have the matrix, we can say by computing what are the ratios of all the molecules that will give rise to reproduction. Now I move a little bit to micelles because they have some advantages. This is just to say that they are very, very strong in catalytic world. And these two, for example, there is one case in which you get a lipid with head group that is a nucleotide, as you can see here. And another one is a tripeptide. And it can be anything you want, and you can do all sorts of things with that. And same is true when it does the game of trying to begin life. So this is another paper that we actually found very few days ago by Colomer, and he does what he calls lipoamino acids, which is something that I've been saying all along, but people said, no, that's not a lipid. We don't have anything like that. Maybe there are some. Okay. So anyway, the idea is that here, just one amino acid can catalyze his in this case, same as for Kate, and with including histidine, and the tyrosine is coming in to become another lipid with an activated fatty acid, a chlorinated fatty acid. Okay, and you get it, it works. So this is really, really nice. There is a lot of things that you can do with micelles that you cannot do with vesicles. This is the real relation in size between this and that. And this big is not bad in this case. Anyway, since I'm getting too late, so I will just say, why don't you read all of that and see, spontaneity, volume ratio, concentrations very high, one molar easily for compounds inside, and so on, diffusing, polarity gradient, which is very important for the catalysis. And they are very similar to globular proteins, because the inside is very apolar and the outside is very polar. And that is very important oftentimes for catalysis. So it's not some like proteins, but very similar to enzymes. Okay, let's continue. This is another real experiment in which this is interesting, mutually catalytic lipid synthesis in a flow cell by Fletcher and colleagues. And what happens is the following. I don't want to go to details, but you end up with, there are many, many vesicles there. And there are 3 different types of vesicles. Actually, I don't know that for sure, but there can be 3 kinds. No, there are 3 kinds, because there are 3 different types of tail. So it turns out, without going to detail, that you get a steady state with homeostatically preserved composition. The most is red, you see, or purple, sorry. Medium is blue, and then green, until you stop the story. When you stop it, then it goes down back. Okay, so this is really, really interesting. It says you can do it, and particularly with micelles. It's good to do experiments on that. There's also something like that. I will skip that for the moment. So we thought quantitative chemistry model showing self-reproduction in micelles, hoping to support future experiments, and also ways by which, if you do it already for micelles, it can then be going into vesicle. Okay, that's very interesting. So, okay, that's enough for that. And this is a very simple explanation of what you need. Homeostatic growth requires net flux in of lipid types in proportion to the present endogenous lipid composition. This, in our simulations, at any point in time, it grows slowly, one at a time, of lipids coming in. And the idea is that you can actually, you can, in the simulation, you can look at the time. At each point in time, there is a different composition because something came in. So we always start with what is already new, and still it works. It's not easy. Okay, so anyway, if this happens, again, you don't have to worry about the physical, at least not for micelles, and by catalytic entry, show here, or by, and again, I have to say that again. Okay. Now, this is really the model. The graded autocatalysis application domain, GARD, a systems chemistry, rigorous computer-simulated kinetic model of a mutually catalytic network. That's what it is. Now, I cannot tell you all about the equation, but it doesn't really matter. First of all, there are as many equations, as many colors, because each of them has a different, what you see here is always the same, but each of them has different properties, so different, it's actually, for each of them is the same. But there are different catalytic effects. So for example, hold on, You cannot see my, hold on, pointer options. Pen, I didn't do any pen so far. No, I don't want pen. I want, what is it, pointer option, highlighter, no, pen, laser pointer, okay. So this is beta JI, one of the elements in the matrix, and you have this, and you can actually compute what will happen. Okay, that's all. So who will go in? That is the one that got, it changes all the time. So the one that gets the best catalytic effects together from several molecules, it can be. It's not only A to B and C to D. It can be also many to one. Anyway, you get the results. Okay, so this is the result. Okay, there is, of course, a micelle and a matrix, and then we go for results. The results looks like it. That's not easy to understand because it's time against time. But what I can say is something like that. Red square indicates 100 compositional similarity over time period, i.e. is self-reproducing. It is a composome. Okay, that's what it's called here. Let me go one more and we'll see something that will explain a little better. Hold on a sec, sorry. Yes, okay. So if you look at blue, just the beginning, it's blue, no composition, no composome. No, there is composition, sorry, but not composome. It doesn't reproduce. And the red is reproducing because you have a lot of time points from about 35 to 60. All along that, they are all red to each other. Red to each other means there is very small difference in composition. Now, these are both, we are talking here about two networks. One, as you started the story, so it has, in this example, 64 lipids, and it gets down to 16. And there is a selection here, between here and there, in the process that goes, let's say, a long time, let's say, from here to here, okay? And this is really interesting because there is some kind of a selection on the molecular background, I would say. Okay, let me go then. There is also metabolic guard, and this means that we can do the almost exact kind of equations, except that we do, instead of beta, we use gamma, which is a beta plus beta. If you do, for example, this plus that with this catalyst gives that, okay? We saw that already. So we use how this interacts with this and this in that. It's one guess that we came up with. Anyway, the idea is it works, and you can also get rich dimer composome. So it's really, really interesting. And not only that, you can get chiral, pure chiral, if you have 50L and 50D, and you run the story, some of the composomes come only blue, which is L, and others only red, and some are racemic at different degrees. So this is really, really interesting. It's interesting because what we are coming up with tells you something that, say, forgot about the idea that you have to work very hard, Miller-Urey type of hard, to get homochirality. Or maybe you can use some strange physics for that. No, you don't need that. It happens with what I say is the beginning of life. That's not less unimportant. Oh, okay. It's not less important. Sorry. You start on our computer with many different initials. Here, and here, and here. Those like the 64, okay? Very, very flat, a lot of molecules of different, many types of molecules, and they don't do much. But it gradually moves, and we were able to prove that it is what is known a dynamic attractor. Actually, the equations that we use say that it should be an attractor. This is unbelievable. It means that the rules of chemistry gets to the point that not only there is a chance that there will be some protocells that reproduce, but it almost needs to happen. And that's really, really nice, if I'm right. Now, if we just throw randomly only 10 to the minus 13 compositions actually is a composome. You see it here, okay? But there are 10 to the 33, it can be 40, it can be 30, it doesn't really matter. But it's a very big number on the entire Earth. So we have, in this particular computation, it is 10 to 20 composomes may form on Earth. And this means that this is composomes, very, very few, but there's still a lot. So about 100 per centimeter cube on the entire ocean. Okay. So here is what I say. We have to believe that all of this is alive in the sense that it reproduces and undergoes Darwinian evolution. And then it becomes more like this. It can be changing of just a few changes in lipid chain, like 2 instead of 1. I show it here. That is 1 in all of these, but here there are beginning to be 2, many of them. So this, because of the Israel-Ashvili rules, it will make more likely a vesicle. So it can move like that, but you can also do fusion of micelles into vesicles, a little bit like horizontal transfer. It's like a gene goes into a bacterium. Here it's a micelle with something interesting on it, getting into a vesicle, and helps it getting better and better capacities to reproduce as a big entity, such as vesicle, with many different kinds of lipids. So this is actually the end, in the sense that I tell you how it all happened. And this is like minus 4 billion, and this is about minus 3.5 billion. So what happened is that this I started first. I should say that micelles can do a lot of, it can generate catalysis and interactions in different ways and so on, much better than vesicles. But it can actually get much more different types of catalysis and types of molecules and so on and so forth. But then it goes to empty. I said empty because you don't want to solve the problem right away. You have to first solve the problem of vesicles that have nothing inside. It's just the vesicle, a lipid, which means just one phase, but it can do all sorts of interesting games, and like the ones that I showed. And then it begins to have things like transporters, but as we know, a dipeptide can be a transporter, so it's not so something. People say transporters. Oh no, this is a very complex 7 transmembrane or 12 transmembrane helices, and that's wrong. We saw it. So the idea is that you begin with something very simple, but it's very important to say that all along, whatever I show here, they should be reproducing and undergoing Darwinian evolution, or else it doesn't work. If they lose their capacity, then they lose the capacity. Those that are there and didn't lose, they will survive. Okay, so we continue to that, and then you begin to do very small, with some molecules that are maybe even a little bigger than before and more complex, and then it makes dimers and trimers of all things. It doesn't have to be proteins. It doesn't have to be. It's really funny. People think that there is only peptides and oligonucleotides and oligosugars, but that's wrong. You can have all sorts of other molecules. So it started like that, but then it narrowed down to what we have to life today. So it gets longer and longer, and some of them are going to be perhaps small peptides. Each of them, like if it's a 10 peptide long, 10 long peptide, it has to be sometimes, or most of the time, an enzyme for each of the putting it together, the two amino acids, to go from 2 to 3 to 3 to 4 and so on. And this happens in bacteria with non-ribosomal peptide synthesis. So it starts like this. And then it becomes bigger and bigger. And then you begin to be big enough. Maybe it can be fold a little bit and get very smart. That's true for RNA as well as peptides and also for others, perhaps. Anyway, we go like that. But what I'm saying is that simple protein and RNA, but this is not yet RNA that can make big proteins. This should go all the way to the point at which we have pre-Luca-protocell. And that must, I mean, if Luca has it, so just a little bit beforehand must have been everything. Protocells, tRNAs, code, akol. This was in Hebrew. Everything. Okay, so this is the story that I have in mind. And I will just say this. Dobrzansky, that said nothing in biology makes sense except in the light of evolution, biology. So people usually say, okay, it's all the way not biology, it's chemistry. I say no. Chemistry can be biology, it can be life. And it's doing, it does the real Darwinian evolution. So it's alive. And I say he would have been happy if I'm correct. Now, when you ask me questions, I'll find out. Thank you. Oh, I have to show one more. This. So this is Roy, who is here on the computer, on computing, and he's the only person that does now Origin of Life with me at Weizmann. But all of these, I talk to them all the time. They are the oldest, a little less, and we get a lot of work together, and sometimes also write papers together, even though they are not with me anymore. So thank you very much.
Thank you. Thank you so much. That was amazing. There are questions in chat. Can you see chat?
Yeah, hold on. Let me get, I want to, okay, I will go here, and then I go to chat, Mr. Chat. Oh, yeah, but it doesn't open. Why doesn't it open? Show chat previews. I don't know what that is. No. Okay, good. All right. So it got it. This is the newest. You have to tell me, Katie.
I think I don't see what you're pointing to, but they're going from the top. So Jose is the first comment.
Okay.
The oldest.
Who is that?
The oldest comment is from Jose. We're all
okay.
a bit of origins researchers here. So that goes in that order.
So Dugard simulations, this is it?
Mm-hmm. Yep.
That's the question?
That's the newest one, yep.
Okay.
That's the newest one.
Oh, that's the newest. So I have to go to the other one.
Yeah.
Up to you, that doesn't really matter.
Is this, no, I said that I want to do first is first. Okay.
Yep.
So this is, hold on. So what's going on here? We're all a bit of origins researchers here building. Good. But I didn't explain the basis of origin of life, right? I mean, okay, just the model. Okay, so this is it. Okay. Do we have to use vessels?
I assume it's vesicles, Doron.
Vessels.
Yeah, it
No,
seems like
maybe
misspelled
vesicles are smaller
vesicles.
than vessels. Okay. Build, okay, get rid of it. Okay, build protocells. Hold on. Do we have to use vesicles, I assume, right?
Mm-hmm.
To build protocells, or do you think, oh, coacervates? Oh, good, good, good. It's an interesting question. I say you can do anything, but am very strong about continuity. We have only lipids now. Now, you can say that I also said that the repertoires changed and so on, but I would say that coacervates are very nice. There are wonderful papers about it. But by the end of the day, they have to move to lipids. Now, on the other hand, if coacervates were more easy to get than vesicles, I would say fine. But usually, it's quite difficult to make coacervates, and they are not very stable, and they sometimes make droplets and so on. And I don't know. I would say it's better to start with lipids. Next. When you study lipid fission, not lipid fission, the vesicle.
Lipid. I mean, I think what she means here is vesicle fission.
Yes, yes, okay. No, because lipid can also change into head and tail, but it didn't. This was not the case. Okay, so vesicle lipids, lipid vesicle fission. Do you consider solid support like minerals? Yes, I consider that there are minerals. I consider that they can make new compounds under some conditions. But I don't see how minerals, although some, there are minerals inside us. That's not the point. The point is that minerals are not as good as micelles, and not as good as membranes, and not as good as proteins and so on. So if there are proteins, but at that point in time, there were no proteins, so I take it back. So the idea is that yes, there are wonderful papers about minerals, but I don't read these papers at all anymore. I'm sorry if maybe there is somebody in the crowd that is doing minerals, so I have to be very careful. But it's not bad. It's just it's less simpler and less makes sense that you start with molecules that are not inorganic. And it's interesting because there are so many papers about this. Same for, yeah, okay. Is there anything else that did anything?
Yep, there are more questions. Next one. Do you see the next one from Melanie?
How does it start?
So the next one, I can read you the next one. It's
Okay.
do guard simulations account for temperature? How would results change with higher and lower temp?
Interesting question. Maybe the person that asks this should tell me what kind of delta T she is talking about. Is it between 0 and 100 or what?
Melanie, if you want to unmute yourself, although people usually don't unmute themselves here, I think, I mean, if I ask that question, I would say I'm curious about the possible prebiotic temperatures. So, anywhere between in the liquid water range. So from a 0 to 100, probably.
Yeah, I would say the following. Temperature is important. That is what is called thermodynamics, okay? The constants change, the catalytic constants change, the delta G is changed, everything. There is always T there in temperature. So the idea is that yes, there can be different conditions, different possibilities, and so on. But my idea is, people tell me something like, what if the vesicle is not permeable to a chemical X? I say, okay, there will be another one that will. This is like, I think about Darwin always. There are always things that didn't work, but there will be some others that will. So if it becomes very hard, I mean, look at what happens here. We had some ice, whole Earth ice, and the same the other way, very, very hot. And some regions in, volcanoes and so on and so forth. Somehow we are still here. So it means that there is a lot of plasticity here. And this is what is so nice about the idea that we are talking about evolution, not just one type. Well, most of the papers do, I just select one type of fatty acid because somebody told me that it is nice, or it is good, or it is possible, or so on. And then we do this experiment, and we add something like that, and we change it. We are talking about the whole planet. The whole planet doesn't care about things like that. If it dies there, it will be alive in another place, survive in another place. Is that a good answer?
It's good to me. The next question is, if you want to perform GARD in experiment, how to add selection pressure so that the system can undergo Darwinian evolution? And that's a good one.
Pressure? There is no pressure. There is just changes in environment. An organism looks it as I think, as pressure. But if we do the experiments, okay, you know what? Let me show you. Can I show you something?
Yeah, I think you can. You're still sharing, so we see your screen.
Okay, all right. So, why does it? No, not stop share. Okay, this one. Okay, these are three worlds. Oops, not this. It's yes, it's this one. So I think the first most is the computing, molecular dynamics. Molecular dynamics is becoming very, very strong. It's basically part of the story of artificial intelligence. And it will probably, some people say that the whole bacterium, molecular-wise, that is every single molecule, water, all that is in the water, the lipids, everything, in a bacterium will be able to compute, so to speak. What we want to do is much simpler, because all we want to do is the following. Take a micelle with some combinations of lipids and let it run. Now it runs in not what is known as molecular dynamics, which is every atom is there, and you know how they touch each other, how they vibrate, everything is there, okay? So the idea is that if you can use this relatively new capacity, we will be able, maybe in 5 or 10 years, to be able to compute this. Compute in the sense that we throw in all the molecules and ask what happens. We started doing something like that. This, what you see here, is something that we did from individual to micelle, okay? We were able to do. This is also something that is known as FACS, fluorescence activated cell sorting. We can have, sometimes it's getting there, that we can do it with a resolution that a single micelle, which is very, very small, in water, or in water, can be sorted in different ways. And this is something that can help very much understand what goes on. And the third is something like I showed you with organocatalysts that do with amino acids here. And I think this is also on Y cells. And I think this can be done maybe by some other cases of, maybe also by molecular dynamics. But I would not worry so much that like in 2 generations, it would be impossible still to see if this is right or wrong. But there are also a lot of experiments that show it already. Next question.
I think that's all the questions that I see in chat right now.
Okay, that's good. That's good. Yeah, you said we want to be about an hour. It's about an hour. And it was a lot of joy for me. Did you also, the questions are also, the questions that were spoken are also, oh wait, but all my answers were also recorded, right?
Yes, everything was
Okay.
recorded.
And if I could get the questions, I will be very happy.
I will send that to you.
Okay, thanks.
I'll send
Thanks
you the
a lot.
chat recording. And thank you so much for a fantastic talk. Thank you for really interesting framing of that problems.
And thank you for
thanks,
inviting me. And I really, really, really appreciate what you're doing, all of you.
Thank you. Thanks, everyone, and have a great rest of your Monday. And Doron, enjoy Greece.
Thank you. Bye-bye.
Bye.